Hello everyone. Welcome to the 10th Public Health Alliance for Genomic Epidemiology webinar. Very glad that you're all here today with us. We're going to have Dr. Robert Beiko today with us on the presentation entitled Different Strategies to Predict Antimicrobial Resistance in Enterococcus. So a brief biography on Dr. Beiko is here with us, and I hope I don't botch your lovely resume. You're our professor and head of the Algorithms and Bioinformatics Research Cluster at the Faculty of Computer Science in Dalhousie University. Your research aims is to understand the microbial diversity and evolution using machine learning, phylogenetics, time series algorithms, and visualization techniques. So very focused on developing tools and pipelines for comprehensively survey genes of mobile genetic elements in bacterial genomes, understanding how these genomes have been shaped by vertical inheritance, recombination, and lateral gene transfer. So very hot topics currently and very in line with Public Health Alliance for Genomic Epidemiology goals. Robert is also co-founder of Dartmouth. Is that how you pronounce it?
Dartmouth, yep.
Dartmouth Ocean Technologies, Inc., developer of environmental DNA sampling devices. This presentation is going to last for about 45 to 50 minutes, if I'm not mistaken. We'll be taking questions at the end. All your questions, please write them down on the chat or in the Q&A box, and I'll be moderating them at the end. Thank you again for being here, and thank you, Robert, for taking the invitation to be with us today.
Thank you very much, Ines, and it's great to see you, and I appreciate your introduction and the organizational efforts of everyone, so it's great to be here. So I've made a terrible mistake because the second slide of this presentation was supposed to acknowledge the fact that this work has been almost entirely done by my PhD student, Ji-In Kim. So she's acknowledged at the end, but I just really wanted to emphasize at the beginning that this is largely her work. So with that in mind, I'm going to be telling you a little bit about machine learning, antimicrobial resistance, and the genus Enterococcus. So it's customary for researchers at Dalhousie University to make a couple of statements regarding historical inequities and the fact that we are working to try and make the situation better. And so first of all, we are located in Mi'kma'ki, which is the ancestral and unceded territory of the Mi'kmaq people. We are all treaty people. We also recognize that African Nova Scotians are distinct people whose histories, legacies, and contributions have enriched that part of Mi'kma'ki, known as Nova Scotia, for over 400 years. So let's talk about Enterococcus. I'm sure most or all of you have heard of Enterococcus, the genus, before. It is closely related to Streptococcus, and there's over 50 named species, as well as a bunch of genomes that kind of fall between the cracks at the moment and are unclassified. As is the case with many pathogens, it kind of hangs out in the intestinal flora a lot, but it's generally pretty rare and pretty innocuous until things start to go sideways. Because in particular, two named species, Enterococcus faecalis and Enterococcus faecium, are really important nosocomial pathogens, right? So when individuals are under other types of stress, then they can turn into serious problems. And they're part of this escape group of pathogens of concern. They can cause a whole range of different conditions, including bacteremia, urinary tract infections, wound infections, et cetera. And in many scenarios, they actually have a very high mortality rate. So these are seriously problematic organisms. Highly tolerant to variation in temperature, and particularly pH. And from the point of view of a genomics lateral gene transfer nerd, one of the things that's exciting about them is that their genomes readily exchange genetic material. They have mobile genetic elements all over the place. And so that makes them really interesting to study, as well as a really high-risk organism, or set of organisms for transmission and emergence of AMR. So speaking of AMR, Enterococcus is almost the poster child for antimicrobial resistance at this point. So I just want to show you a figure from a paper by a colleague of mine, Rahat Zahir, and her collaborators, published in 2020, looking at, for the province of Alberta, Canada, across a range of different habitats, the incidence and types of antimicrobial resistance. And so what you see here, starting with one species, Enterococcus faecalis, across several different habitat types from agricultural all the way through to clinical, the number of different resistance types, right? So how many antimicrobial agents are you resistant to? And you can see that from blue, which is 0, to red, which is getting up towards 7, there are different degrees of resistance. And then you can look at the specific profiles and see which types of antimicrobial agents tend to be commonly associated with resistance. And so one thing you may notice is that there's not a lot of black, right? So vancomycin is the black antimicrobial here. And Enterococcus faecalis is not typically associated with vancomycin resistance, but you can see a lot of other stuff, tetracyclines, erythromycin, and to a lesser degree, other antimicrobials as well. When we look at Ephesium, so its sister species, we can see a lot more in terms of, especially if we look at the clinic, we tend to see more vancomycin resistance. That comes up a lot, as well as some other stuff like fluoroquinolones, beta-lactams, and then again, familiar stuff like tetracyclines and erythromycin. And so Enterococcus is resistant to a lot of things, and this is obviously a huge concern. The focus in this presentation is going to be on these three: erythromycin, one tetracycline in particular, doxycycline, and then vancomycin. This is just a couple of other illustrations of the resistance profile. So you can see that on the left, we're looking at Enterococcus faecalis, and these curves are relatively flat over time if you look at things like VAN and aminoglycosides and aminopenicillins. But again, this is faecalis, which tends not to have the vancomycin resistance. On the right, you can see some global information about resistance. Obviously, these are difficult to compare across countries. The main message here is that it is found in many different corners of the globe. And I think one really important point to emphasize is that a lot of the last resort antibiotics, the ones you try when you've gone through many of the others, is increasing quite dramatically. And so from a point where things like linezolid and daptomycin worked fairly well, we're starting to see rapid emergence of resistance to these. So what makes Enterococcus resistant? So there's a few different mechanisms of resistance here. And some of the basic ones, what's called the intrinsic resistance, like antimicrobials that tend not to work very well from the get-go. Enterococcus has certain properties like a thicker cell wall structure, certain types of inactivating enzymes and efflux pumps that push antimicrobials out of the cell. So this is intrinsic. But then on top of this, acquired resistance can come in several different forms. So as I said, Enterococcus is really good at transmitting genetic information, particularly through mobile genetic elements. And these include things like plasmids and transposons. And so vancomycin resistance is often associated with plasmids, not always, but quite often. And then there are these different elements such as integrative conjugative elements, which can pop quite easily into the main chromosome or indeed a plasmid, as well as transmit themselves between cells, such as a plasmid would typically do. And one sort of common example of this ICE element is TN916, which has all of the genes necessary to do this mechanical stuff, and often has a cargo gene for tetracycline resistance, and sometimes carries other cargo genes as well. Insertion sequences, transposases, really support the rapid co-integration and reshuffling of all these mobile genetic elements. And so you often, when you look, you see different combinations of these things: plasmids, transposases, ICE elements, and so on in various combinations. In addition to this, there's also de novo mutations that emerge. So these can be protective against certain types of antimicrobials. Daptomycin is often conferred through changes in cell wall structure based on genes that are already present. A couple of examples of mobile genetic elements. So this is an example of a plasmid, just over 42,000 base pairs, that's found in multiple isolates of Enterococcus faecium. And it's a bit small there, but long story short, It's got all of the genes necessary for conjugation. It's got various antimicrobial resistance genes and other mobile genetic elements like transposases. Lower left, you can see a series of genes in green. These all confer resistance to vancomycin. On the right-hand side, this is that TN916 integrative conjugative element, or ICE. And again, the canonical ICE has all of the elements necessary to recombine into the genome, excise itself from the genome, as well as carry out conjugative transfer. When you look at actual genomes, you often see degraded versions of TN916, and these are either non-functional. They might still have the accessory gene. There's that tetracycline resistance gene. They might not be able to excise. They might not be able to conjugate. In some cases, they can be transactivated, so something else in the cell might still be able to help trigger the recombination events. So if we want to think about how to predict the resistance profile of Enterococcus, we can say, well, what do we have, right? What are our basic assets? And so one is that there are lots and lots of genome sequences, right? There are over, I think, 12,000 genome sequences of the genus Enterococcus available now, and lots of them are short read assemblies, but you can still have annotated genes and mobile genetic elements. And for a subset of these, we have actual assessments of antimicrobial resistance. I'll say more about that a bit later, but for many isolates, we have a genome sequence and we have a profile that says it's resistant to the following antimicrobials and susceptible to others. The key challenges to all of this, one is the issue of false positives. So you can take a set of genomes, annotate all of the genes in those genomes and say, aha, here is a gene that I know confers antimicrobial resistance. But you don't necessarily know that it's functional in the cell. Maybe there are inactivating mutations. Maybe there's a promoter mutation and the thing's never expressed or only expressed under a restricted set of conditions. The flip side of that is false negatives, where you fail to identify specific genes, you fail to annotate specific mobile genetic elements. And this is a known problem in short read assemblies because particularly the genes and MGEs we're most interested in have lots of repetitive sequences that tend to break short read aligners. Another issue is that many resistance types are common, and those are the ones I'll be talking about a bit later, but some of these emerging ones will still be fairly rare. And so if you have a set of, let's say, 500 genomes, and only 5 of them are resistant to a particular antibiotic, then using any sort of model to try and do classification is going to be really tough. Both because training a model on such a small data set is going to be challenging, but also because when they're rare, it's a higher likelihood that the mechanisms haven't yet been elucidated very well, which means that any prior knowledge is going to be difficult to inject into your training. So why are we going to use machine learning for this? Well, I mean, obviously, we're interested in feeding in genome sequences and getting accuracy, right? How well can we predict the resistance of a given genome to erythromycin or vancomycin or whatever, right? Magic numbers. Machine learning is great in that you can take a series of features and represent and capture complex interactions among them, right? So and relationships or relationships, those kinds of things can be modeled quite well or using machine learning methods. Another issue is, or another benefit of these is that in cases where you have maybe multiple resistance mechanisms for the same antibiotic, right? So VAN is a great example. There's multiple ways to be resistant to vancomycin. A machine learning method might be better at actually learning the rules to hesitate to use the word explain, but to accurately classify more than one category of resistance type. They can highlight key features. So you can look through all of the features you've generated, genes, mobile genetic elements, and identify the ones that are important, relevant for classification. Now, of course, there's the standard caveat of causation versus correlation, but these can all be informative in different ways. So these can give you gene lists, and we all know and love the interpretation of gene lists. One thing that's really fun is that you can follow up your features more closely by looking at things like gene order, by looking at things like evolutionary history to see whether there's evidence of lateral gene transfer. It's also interesting because you almost never get 100% accuracy. That's not how biology works. But you can take a look at the misclassifications if your model's pretty good and say, it got 95% of the cases right. What's the deal with the 5% it didn't get right? Are these weird artifacts? Did the assembly just fail and you didn't annotate a key plasmid or a key resistance gene? Or is there something more kind of biological going on? Are there intermediate patterns of resistance, for example? And so one thing that's really interesting to do is try and tease apart these stories for the cases where we get wrong. Okay, so I'm going to tell you about a project, and this again is the work that she has done in collaboration with some colleagues at the Agriculture, Agri-Food Canada agency. So here's the data set we looked at. We have just over 640 isolate genomes total between Faecium and Faecalis, roughly balanced between the two. These are all collected by our collaborators at AAFC in Alberta. They were all subjected to short read sequencing and assembly, and they cover a range of habitats. So we have some clinical isolates, we also have some agriculturally associated, and we have some from surface water or agricultural runoff. As I mentioned before, we have this other information as well. We have resistance profiles. And so I suspect many of you are already familiar with this, but the basic idea, one of the more common ways to assess resistance is to try and grow the your bacterium, your pathogen of interest, in the presence of different types of antibiotics, and then look at the degree to which different antibiotics can impede the growth of your organism. And so on the right, you see what's called a disc diffusion assay. And you can see sort of the yellow parts are the parts of the Petri dish where the organism of interest has been able to grow. Each of those white discs contains a different antibiotic. And you can see that in some cases, the radius around the white disc is very small, right? So the bacterium is able to grow up pretty much right to the edges of the disc. If it can do this, it's because it's resistant to that antibiotic. It's having no impact on its growth, right? So that would be an example of resistance. If we look here, though, the antibiotic's been quite effective at clearing an entire part of the dish of the bacteria of interest, of concern. So this is a quantitative score, and we can take these quantitative scores, typically expressed as the zone of inhibition size in millimeters, and then we can take our isolates and plot them along this gradient. So here what you're seeing, x-axis is this zone of inhibition size. Each row is just a different isolate genome, and you can see that genomes have different associated zones of inhibition. To turn this into a categorical classification, there are different standards established by a couple of organizations to say if the zone of inhibition is greater than X, then we have something that's susceptible to a given antibiotic. And so in the case of vancomycin, you can see that we have these thresholds, and these are used to define three categories. On the left, you can see the ones that are highly inhibited, that are not inhibited, and these are the resistant isolates. To the right, you can see the ones that are susceptible, and in the middle, there's this gray zone where they're identified as intermediate. So these are the three categories we're looking at. So here's what we get when we look at the overall resistance profiles, when we turn these zones of inhibition into actual categorical predictions. And so on the left is E. faecium, on the right is E. faecalis. Green is resistant, I is intermediate, and orange is intermediate, and purple is susceptible. And you can see that the two species have somewhat different profiles. This is particularly true with vancomycin resistance. So you can see that on the left, 32.5% of the E. faecium isolates are vancomycin resistant, whereas on the right, only 1.1% of E. faecalis isolates are resistant. There are some that fall into that intermediate category, but in general, and this is generally true. E. faecalis shows a lot less resistant to vancomycin than its sister species, E. faecium. But in terms of machine learning classification, these categories are ones we can work with, right? So we have enough susceptible, we have enough resistant cases that we can try to build models to classify the two. This is just drilling down a little bit more, not super critical for the rest of the presentation, but just to show you that we have stratified these in terms of their total overall resistance profile. So the upset plot shows for each antimicrobial, vancomycin, erythromycin, doxycycline, the number that are susceptible, resistant, or intermediate, and then each x-axis column is the number of isolates of faecalis and faecium that match that resistance profile. So for example, we can look at these three, which are the most common categories. These are all sensitive to vancomycin, resistant to erythromycin, and then have some doxycycline property, whether it's sensitive, resistant, or intermediate. And so you can look for different specific categories if you're interested in multi-drug resistance, rather than looking at them one at a time. You can also ask questions like, how many isolates are resistant to all three? And the answer is 8. They're all faecium, not surprisingly, because of that vancomycin story. And then you can say how many are sensitive to all three antibiotics, and that's 36, roughly balanced between the two species. So that's the AMR side, that's the resistance story. This is what we're trying to predict. What about the features? What is the actual basis for prediction? How do we take our genomes and turn them into feature representations that we can use? And so there's a few strategies we use. The first one is based on a highly curated data set of antimicrobial resistance genes. So this is the Comprehensive Antibiotic Resistance Database, or CARD, and there's a software package associated with CARD called the Resistance Gene Identifier, RGI, which will take a genome, compare against all of the manually curated, experimentally validated genes that are in CARD, and make predictions about them. And the RGI stratifies the hits into 3 categories. A hit is perfect if it's a perfect match to one of the experimentally validated genes in the reference database. It's a strict hit if it's very, very similar and has a high probability of being real. Below that, below a threshold of, say, 97% identity, depending on the antibiotic, you get what are called loose hits. These are ones that are homologous and highly similar, but not similar enough to meet that threshold. And so people typically, when they're using the RGI, will focus on the perfect and the strict hits. This is just an example of what you get, a visual example. If you take one particular isolate genome of E. faecium and feed both its chromosome and its plasmid through RGI, and you get these different colors. Perfect is green, yellow is strict, and orange is loose. You can see in this case that there are no loose hits at all. Both the chromosome and the plasmid have perfect hits, and it's maybe a little bit too small to read, but on the plasmid, you see a whole series. All of the perfect hits are vancomycin resistance genes. So this is a way to profile the genome and say, these genomes have this vancomycin resistance gene as predicted by the RGI. In addition to this very sort of highly curated, highly targeted approach, we also took the genomes, annotated everything we could using a set of 3 different annotation tools, that's PROCA, RAS, TK, and BACTA, and then used a program called Rory to take the gene predictions of the genomes and construct sets of them. So just like with RGI, you can say, this genome has Van A, the Van A gene, this one doesn't, this one does, this one does, and have like a presence-absence profile of Van A genes. Here, the Rory software tool does this for everything that's annotated in the genome. And so looking back for a second, RGI assigned across the entire data set a total of 62 different genes. Whereas if we look at the pangenomes, you can see that we have a total of just under 30,000 for the PROCA and BACTA annotation tools, and almost 35,000 for the RAS-TK. So this is a much, much larger feature set, most of which is obviously going to be irrelevant, but this gives us the opportunity to discover things that aren't present in the card that may either be causal or may correlate with the phenotypic profile of the isolate genomes. So that's genes. We have the highly curated RGI approach, and we have the much more broad pan-genome approach. We did one more thing. As I mentioned, we know that there are lots of plasmids in Enterococcus faecalis and faecium. These often carry resistance determinants. And so we took our genomes and we did a plasmid-specific prediction approach. To do this, we used a package called MobSuite. Long story short for MobSuite, it takes reference plasmids from NCBI, clusters them based on their nucleotide similarity, and then takes your contigs from your assemblies, compares them against those reference clusters. And if there's enough similarity between the two, it'll say, this contig from your sequence genome, from your assembly, is a good match to some plasmid cluster that's been previously built by MobSuite from that NCBI reference. And so when we did this, when we took our over 640 isolate genomes of Enterococcus, MobSuite predicted or identified a total of 206 plasmid clusters. Some of these were present only once in our set of 640 genomes, so not so useful, but other ones were much more common. Okay, so sometimes it's not the best idea to feed 35,000 different features into a classifier, depending on your choice of classifier. In addition to this, a classifier can give you prediction accuracy, but it's often good to take different approaches to get a much more explicit, detailed sense of how well your features relate to the phenotype you're trying to predict. And so in addition to the feature construction and the actual classification, we also use different techniques for feature ranking and selection. The one that I'm going to be talking about most today is something called the HSIC block Lasso. And so I'm not going to get into detail, but the basic idea is that intuitively, useful features, whether they're genes or plasmids in this case, should have strong associations with the phenotype of interest, right? So very intuitively, if all of your vancomycin-resistant isolates have gene X, and none of your vancomycin-susceptible isolates have gene X, then that's a pretty good association. That's trivial, and there are more sophisticated approaches we need to take to do this, such as this HSIC distance, but the basic idea is the same. Let's look at our features, find strong associations. Those are our most promising candidates. What the Lasso method does is it takes all of these features and looks for constrained sets of them that are really good in association. So it's not just going one at a time, it's looking at the entire feature set and saying, what combination of these with what associated weightings are really good at associating with the phenotype? So what it'll do, it'll have a penalty that tries to exclude most of the features, the ones that are useless, and then assign weighting. So if something associates really well with the phenotype of interest, it gets a higher weighting, gets a quantitative weighting. So the Lasso really is good at giving you a big comprehensive picture of everything. The other thing we used, the other measurement we used, was a simple Pearson correlation. This allows us to look one at a time at our different features and say, independently of everything else, how well does this feature associate with the phenotype of interest, right? So Lasso is big picture, everything considered at once. Pearson correlation is one at a time. We tried 3 different machine learning methods, and full disclosure, we're not as interested in the choice of machine learning methods. So we just tried a few to make sure that, you know, the shortcomings of one wouldn't impact our accuracy overall. So we ran logistic regression, random forest gradient boosting, and did 5-fold cross-validation. So the accuracy scores I'm going to show you are, with the 5-fold cross-validation, just the average across all of those validation sets. We can get into that. I'm not going to say much more about that, but if there are specific questions about that, happy to address them in the Q&A session. Okay, so that's what we did. We have our genotype information encoded as resistance genes, encoded as pangenomes, encoded as plasmids. On the other side, we have all of our phenotype information, our resistance profiles, right? Again, those 3 categories: susceptible, resistant, and intermediate. One thing I forgot to mention is that all of the training and testing, all the accuracy scores, were done only on the resistant and the susceptible groups. We did not include the intermediate in any of the quantitative evaluations. We do look at them later on, but since we didn't know where to put them, we set them aside for the scores that I'm about to show you. So just to walk you through the 3 different antibiotic classes we looked at. So the first one is vancomycin. And vancomycin is relatively easy. And the reason for that is because there are certain widespread sets of genes that are known to confer vancomycin resistance. Particularly in this set of Alberta genomes, we know in addition to that, that it's really like one specific mechanism, the Van-A operon. So this gives the feature selection methods and the classifiers something really good to grab onto, which has the strong potential to lead to perfect or near perfect accuracy. And that's what we see. So you look at the top line and the RGI, this is again that curated database of specifically genes known to confer antimicrobial resistance. We got 99.2% accuracy, so almost perfect prediction accuracy. Those 3 pangenome methods, PROCA, RAS, TK, and BACTA, all gave us 98 to 98.5% accuracy. One thing that was interesting was when we used MobSuite, this plasmid prediction tool, and just fed the plasmids to the classifier, so no explicit information about Van-A, any other genes, we still got 96.7% accuracy. So looking at these annotated plasmids, in many cases, was enough to give us this predictive information. What are the key features? So again, this is that weighting that we can get from the HSIC block Lasso. And oh look, the ones with the highest weightings all have Van at the beginning, and that's because they're all part of this operon that confers vancomycin resistance. There's a few that are further down, including another vancomycin resistance gene, but this is really dominated by those top 5. And if we look at, so that was the RGI. If we look at the pangenome, so again, this is in the case of BACTA, almost 30,000, and in the case of RAS-TK, almost 35,000. I'm not showing you PROCA because it's quite similar to BACTA. So this is 2 of the 3 methods we used. When we look at the key features, out of all of these different genes that the classifier had access to, the most highly ranked ones, again, Van X, Van H, Van A, Van R. So not too surprising here. And the main differences when you look at the 2 lists are not so much in the genes themselves as they are in the names that are assigned to the genes. So once again, we almost refer to this as our positive control because the accuracy is so high, we know what to expect, and we get what we expect. But the plasmid story is kind of interesting because these Van resistance genes aren't necessarily just carried on a single type of plasmid. They can move around. And so there's multiple different plasmids that might show up in our feature selection, our feature ranking. And that's kind of what we see. And so on the left-hand side of this table are the identifiers assigned by MobSuite, right? So plasmid cluster AC733, AE577. These have no intrinsic meaning, but with these clusters, we can take the ones that are highly ranked and look at NCBI and say, which plasmids are these actually referring to? And so the top ranked one, AC733, is actually quite a large plasmid, 216,000 nucleotides, 236 proteins. The canonical NCBI plasmid was from a vancomycin-resistant Enterococcus faecium. Does it have the Van genes? No, actually it doesn't. So that's kind of interesting. Turns out that if you look at that genome, the Van genes are found on a different plasmid. So here's an example of something that may be more correlative than causal, but it immediately suggests some interesting evolutionary dynamic between the different replicons in that genome and presumably in other ones as well. So that's kind of a surprising top result. The second result, this AE577, is also kind of surprising because it's only 3,000 nucleotides and it has exactly 2 proteins. Also from a VRE isolate, but no Van genes. Obviously, if it's got 2 genes or 2 proteins, then given that it needs to copy itself, there's not much room for anything else. So there's not too much we can say functionally about this plasmid, but one thing we can look at is the taxonomic range of this plasmid. And in this case, we see that identical or nearly identical plasmids are found in very distantly related organisms. So Enterococcus is a gram-positive, whereas Klebsiella is a gram-negative enteric, so very distantly related, Acinetobacter. And so these are super, super broad host range plasmids. So again, no Van genes, but something interesting going on. The third one is where we start to get a bit happier. Well, sort of, right? AB173, 40,000 nucleotides, 34 proteins. Does it have the Van genes? Yes, it does. So this one makes a lot of sense. It's from the NCBI plasmids are from VRE isolates, and these ones actually have the Van genes. So great. The fourth one is kind of like the second one, except it's even smaller, but it also has a broad host range. So for example, it's also found in E. coli. And then finally, the fifth one, also kind of what you might expect to see. Plasmid, 40 to 60 kb, 64-ish proteins, and then the entire suite of Van genes. Okay, so that's the Van story. What about other antibiotics? So let's look at doxycycline. And so doxy, the mechanisms are not quite as singular as vancomycin. So there's a few different resistance mechanisms, and this one may be a bit more difficult to predict. And it turns out, yes, it is. Still above 90% in most cases. So anything involving gene level predictions, we get that reasonably high accuracy, 91.8% to 93.3%. Kind of like vancomycin, we still see higher accuracy with that curated data set. So that's kind of interesting. The plasmids, we don't do so well, 83.4%. This is not a huge surprise because although the doxy resistance genes can sometimes be found on plasmids, often they're not. So we don't necessarily expect plasmids to give us super mega awesome classification accuracy. What happens when we look at the RGI features? Well, this one is pretty unambiguous, right? So TET-M, which is obviously conferring resistant to tetracyclines, like doxycycline, is the only one that stands out with a decent coefficient from the HSIC block Lasso. So that's fine. But notice that the ones to the right, the ones with really, really, really low coefficients, many of them, or almost all of them, also confer resistance. So we see Van genes, we see ERMB, which is erythromycin, we see other TET genes. So even though their weightings are really small, they're still in the antimicrobial resistance space. It's not just random stuff that's getting ranked. So that's pretty interesting. To some, I mean, this is a subjective statement, but I'll just say it anyway, even more interesting is what happens when we look at the pangenome. Because when we look at the pangenome, we actually don't really get TET-M as one of the top results. What do we get? We actually get genes associated with that TN916 transposon, or ICE element that I mentioned. And so in this case, we're not getting the causal gene, we're getting the genes that are characteristic of the mobile genetic element that often carries this gene. And so these are not causal in terms of the resistance mechanism, but they are really important in how this resistance moves around. Okay, so this again, correlation versus causation, but super interesting that this is what we turn up. Now notice that we predicted plasmids. We didn't try to predict ice elements. And one of the big reasons for this is because the naming conventions of ice elements are really, really tricky, and very, very similar ice elements can have completely different names. So thinking of this in terms of clearly named and delineated features is really hard. So what we tend to do instead is just look at individual genes and say, do these associate with TN916 and closely related ice elements? Okay, third one, erythromycin resistance. How about this? Well, in this case, the accuracies are fairly reminiscent when we look at genes of the doxycycline example. The plasmid accuracy is actually a bit higher in this case. Lower than vancomycin, higher than doxy. What happens when we look at the key features? Well, surprise, surprise, surprise. Erythromycin is right at the top. Again, we see a series, sort of a constellation of other resistance genes. This is maybe not super surprising because when you start to look at a lot of mobile genetic elements, plasmids often carry genes, like a single plasmid will often carry genes that confer resistance to 3 or more antibiotics. So it's not necessarily surprising that we see others rising to the top as well. But again, erythromycin resistance, and there's that key erythromycin resistance gene. But again, when we look at the key features from the pangenome, we don't see ERMB or ERMC, right? We see other things like sugar, arabinose metabolism, enzymes that can influence cell wall composition. So there's maybe a pathway to functional information here, but this is not a story we've completed yet because we're still looking at this sort of functional associations. We're also still looking at genomic context to see whether we have these genes in close proximity to the actual resistant determinant genes. Okay. So that's the classification story, the accuracy and key features. What about misclassifications? This is something I mentioned earlier on. So what happens? Can we look at these and ask questions about why things tend to be misclassified? So one interesting question to ask is there a relationship between the quantitative score, this is that zone of inhibition size, and the propensity for misclassification? Basically, what this means is you can have susceptible isolates that are more susceptible or less susceptible, right? And the less susceptible ones will be closer to that breakpoint that I mentioned before. And so you might think that the ones that are closer to the middle, that are closer to the breakpoint, might be more difficult to classify. And so G asked this question and said, is there a relationship between how sort of, the zone of inhibition size and the tendency to be misclassified? And long story short, no, there isn't. Things that are really, really, really susceptible are just as likely to be misclassified as things that are only slightly susceptible. We can also ask really irritating bioinformatics questions. Again, so sorry, these figures are so small, but basically what you're looking at here is every point represents a single genome, and we have the x-axis showing the number of misclassifications. So everything in blue is isolate genomes that were never misclassified. Okay, so these are always correctly classified. We have 4 sets of 3, and each of these sets corresponds to a different evaluation of assembly quality, right? Because one might suspect, especially if we're talking about plasmids, that misclassified cases might be misclassified because we just missed something. That's that false negative argument. And so we can ask questions like, do misclassified genomes have more contigs than correctly classified genomes? Do misclassified genomes have shorter assemblies? Do they have smaller, largest contigs? Do they have poorer N50s? So we can take any measure of assembly quality, compare the averages between correctly classified and misclassified, and ask the question, is there a significant difference between the two? Because if there is, then maybe some of this misclassification is artifactual, but no, it isn't. So we, in no case at all, did we see any connection between assembly artifacts or assembly properties, and the tendency for isolates to be misclassified. Okay, so one thing, and I apologize I don't have a figure for this, but then, as I mentioned before, all of the training and testing was done on resistant and susceptible isolates. We set aside all of these intermediate ones so that once the model was trained, we could come back and say, dear model, what do you think? Right? We don't know the right answer, and to some extent there isn't a right answer, but it's interesting to ask the question because when we know which features are most important, we can look at those intermediate cases and say, do they have those features? And the model tended to classify most intermediate, not all, most intermediate cases as resistant. This means that the features from those genomes, from those corresponding genomes, are the ones that are used to classify resistant isolates are present, right? So in the case of vancomycin resistant, we looked at the intermediate cases, and nearly all of them had the key vancomycin resistance genes. Now, why are they not actually phenotyping as resistant? Well, there's a few possible reasons, right? One is just variation in experimental characterization. Another is the expression levels might be lower, right? Maybe in some isolate genomes, they're just not producing as much of the vancomycin resistance genes. Maybe there are mutations either in the VAN genes or elsewhere in the genome that impact the efficacy of resistance. So there's all sorts of possibilities, but it's interesting to know that at least the potential is there. Okay, so let's bring this home. Questions and next steps. So there's a bunch of big questions that remain. One is this obvious question of correlation versus causation. And a lot of the examples I showed you, particularly with the pangenome information, and also the RGI, right? We saw correlated stuff that clearly wasn't causal. But certainly with the pangenome, we identified a bunch of associated genes that are not causal, but let's say are associated with the same mobile genetic element, right? But that correlation information can still tell us really interesting things about the evolutionary mechanisms, about how these things move around, as well as functions, molecular functions that may be associated with, even if they're not directly causal, of resistance. As I mentioned earlier, we didn't really look into multiple mechanisms of resistance. So in this case, VAN-A was really the dominant mode of resistance. Another example is the VAN-B operon, which is homologous with VAN-A, but the sequences are dissimilar enough that they're treated as separate entities. And so in this data set, we didn't have any of that. In previous work, we have done work with VAN-B in terms of mapping it, doing associations. One really interesting and important difference is that VAN-A is typically plasmid associated, VAN-B is often not. Okay, so this will influence the types of results we get when we do profiling of plasmids and we look at chromosomal genes. Our accuracy scores are inflated. One of the reasons for this, a really important one, is the phylogenetic correlation, right? So as I mentioned, we did cross-validation. So you train on this part of the data and evaluate your model on that part of the data and do that several times. But one issue, especially when you have closely related isolates, which we do, is that your training set and your validation set might have nearly identical genomes. And so in some ways, it's not really a fair test. And there's different ways to compensate for this. A simple one is to say, if this thing is in the training set, please ensure that nothing that's really, really, really similar to this is in the test set, because otherwise we're letting the classifier cheat. Another interesting question is our definitions of genes and mobile genetic elements. I already showed you this for the TN916, where there's a whole range of differently named ice elements that are actually really, really, really similar. The same is true for plasmids, right? It's not like this group of plasmids and that group of plasmids are completely distinct. And so if we adjust our slider a little bit and say, no, this set of related plasmids is one group versus no, maybe we should split it into two different groups, we'll probably see an impact on accuracy. And so one thing we can try is just changing our definitions a little bit and seeing what happens. Maybe this broad group of plasmids is really good at classification, but when we really drill down, it's actually this subset that's defining it, and the others are just kind of along for the ride. Okay, so summary, the classifications, again, subjective statement, highly accurate, but we still have all these false positives and false negatives that we would really, really like to understand. I already just mentioned this issue about inflation, so we need to think phylogenetically. And then we have no evidence whatsoever that there's bioinformatics nonsense that's dropping our accuracy scores. Last thing, I just want to put in a plug for a software pipeline we're developing at AT, which is really meant to do comprehensive annotation, pangenome inference, phylogenomics, and then evolutionary analysis of genomes. So I showed you pangenome, I showed you plasmids, I showed you RGI, but we're using AT now to generate a much broader range of annotations, including things like prophages, genomic islands, metal resistance genes. And so this is going to give us a much richer data set to draw on for classification purposes. And again, when we identify interesting features, the evolutionary part will help us put this into context of things like transmission risk. So it just remains for me to thank the people who contributed to this work. There's G upper left. Again, this work is largely done by her. And then we have our collaborators at AAFC who collected and characterized the data set. So thank you very much. I guess we have a couple of minutes for questions, and yeah, appreciate everyone's attention.
Thank you so much. We do have a couple of questions. I'm going to start with the latest one that I think is more direct. How did you determine the TN916 associated genes? And yes. We can still see your screen, by the way.
I'm sorry?
We can still see your screen. I don't know if that's good.
Yeah, can you see me as well, or just my screen?
I can see you.
Okay, I'll just leave. Okay, yeah, I'll shut it down then. I will stop. Cool. So yeah, it's tricky, but the associations there were determined actually by mapping to contigs, right? So if you look, you can identify parts of the genome that do have these conjugation genes, that do have these recombination genes. And so we were able to identify parts of the genome where these features map to, and they all map to very similar locations, right? And when we looked at it, it was clearly TN916 or something very much like it.
Thank you so much. We have, it's more like a comment and a question from John. The addition of new genes to reference plasmid structures could be allowed to miss critical information. Database must be permanently updated. It could be a solution. Any comment?
Oh, yes. Yeah, I completely agree. And this is an issue, you know, because we kind of have different strata of prediction approaches here, right? So like CARD, RGI, highly curated. And so typically when you get a perfect or strict match, it's real, right? You might miss stuff either because your threshold is not set correctly or because there's stuff that's missing from the database. Again, possibly due to new mechanisms that either have not been discovered or haven't propagated in the literature to the point where they've been added to the card. For plasmids, we are absolutely dependent on the strength of the reference database. So, you know, MobSuite is really built with gram-negative enterics in mind. And so when we applied it to Enterococcus, we're really, if not breaking new ground, certainly getting outside a little bit of the use case. And so it's interesting that we're able to find a lot of very convincing plasmids, but I am completely confident that there's stuff we're missing, both because there's stuff that's not in the database and because short read assembly is not great for plasmid recovery.
And any reason why you went with short reads instead of going with the compromise of trying to do long read sequencing? It has lower accuracy on the base calling, but the recovery of plasmids and repetitive sequences like transposon is higher.
Mm-hmm. Yeah, so the short read genomes were kind of existing data that had already been assessed. But one thing we did was once we characterized these genomes, we identified a subset that were diverse in terms of their habitat of origin, in terms of the mobile genetic elements they seem to contain, in terms of their phylogeny. And we said, okay, let's take about 60 Faecium and 60 Faecalis and do hybrid sequencing. So we did that. We have those genomes. We just haven't integrated them into the framework yet.
Awesome. Can't wait to see those results.
Yeah, it's interesting.
And
Although the hybrid assemblies were kind of disappointing because, and this is
really?
something like I talked to Enterococcus people, they're like, yeah, nanopore is not a perfect solution to this problem. People often use PHA4GE just because they get better results. But,
Yeah,
in the worst case, our hybrid assemblies still had like 200 contigs. And
yeah,
this
that's
is
a
not
lot.
a rocket effort. So yeah.
Last question. The pipeline you showed at the end, a read, can it be used for both long read and short read data?
Yep. So the answer is there are two different entry points. If you want Aditi to assemble your genomes, we implement or we've adopted Unicycler. So, and we've typically just used it for short reads. But we also have an entry point that we tend to use more often, which is just here are the assembled genomes which have already been generated by someone else or through some other software, and we just start at the annotation step and run from there. Because we do have one mechanism, we do have one method for assembly, but everyone has their favorite, and people come with MySeq or HiSeq or Nanopore or PacBio, whatever, and we just can't support all of those. So we can say, we've got one way to do it if you want, but feel free to run it yourself and then bring it to the pipeline.
And as you showed your results, the assembly metrics that did not affect that much your modeling results.
Yeah.
So we're at the top of the hour. Actually, we're 2 minutes past. I would like to thank you so much for being here today. We've answered all our questions, and I have no further questions for you. So thank you very much.
Okay.
I don't know if the Secretariat has anything they would like to say here at the end. Jamie? Jamie says it's all good. So I'll terminate this session for today. Thank you so much.
Perfect.